In this video, I will be reviewing the key features on the structure of DNA. As presented in this picture, DNA is commonly analogized as a spiral ladder, but it is important that it is right-handed, meaning a counterclockwise rotation to the right. The structure was first founded and analyzed by Watson and Crick. DNA consists of two anti-parallel strands, with each strand composed of a sequence of nucleotides that are unique to that particular organism. A nucleotide consists of a base, a sugar, and a phosphate group. The carbons on the sugar are numbered 1 through 5. The primary carbon is the one that bonds to the base, where water breaks off in this process. The secondary carbon differs between ribose and deoxyribose, where in ribose there is an OH instead of an H. Ribose is used in RNA. The 3' carbon bonds with the next 5' carbon, and the 5' carbon bonds with the phosphate group through a phosphodiester bond in order to make this phosphate backbone. This backbone is the hydrophilic part of the DNA structure, which leads to its solubility in water. The nitrogenous bases, on the other hand, are hydrophobic, which repels water, and allows the bases to stack. This has been found to be the most important stabilizing factor within the DNA molecule. The bases are planar, while the phosphate backbone is perpendicular to that plane, which makes it look like an uppercase L. Since the two strands run anti-parallel to each other, when base stacking occurs, one nucleotide's formation is opposite of its complement, which causes major and minor grooves in the DNA structure. As the backbone is represented as the lanyard, the grooves can be seen where the major groove is the wider separation and the minor groove is the smaller separation of strands. There are 4 bases whose structure is split into 2 groups, purines and pyrimidines. Purines consist of adenine and guanine, which are fused bicyclic structures, while the pyrimidines consist of cytosine, thymine, and uracil, which are in a cyclic structure. In these structures, the black molecules represent carbons, the blue and purple molecules represent nitrogens, hence why they're called nitrogenous bases, and the white molecules are hydrogens, while the red molecules are oxygen. Each of these bases has its complementary pair. Adenine binds with thymine and guanine binds with cytosine, but first we must rotate thymine and cytosine in order for the complementary bases to form hydrogen bonding between them. But what about uracil? In RNA, guanine and cytosine still bind with one another, but instead of thymine, adenine binds with uracil. As shown in the illustration above, there are 3 hydrogen bonds that link guanine and cytosine together, while there are only 2 hydrogen bonds that hold adenine and thymine together. Again, the white molecules are the hydrogen bonds coming off of the bases. Though hydrogen bonding is typically pretty strong when it comes to intermolecular forces, when it comes to the stability of DNA, base stacking has more of an influence. Here are some images that show DNA as being left-handed, which is incorrect. You can identify left-handed DNA by its clockwise rotation around the central axis.